You know what, I'll come forward a bit. We built this whole new stage just for today. There we go. Wow, Cellular Dance. So, you all ready for a science lesson? We've got some music too, and some dancing, I hear. So, I've got this video up here, and just out of interest, raise your hand if you know what this is or have seen it before. Not enough of you. We're looking at, obviously, something from the cellular world. They build our lives, and they also changed our lives for the better. 2020, 2021, 2022. Cellular Dance was first performed in 2019, and then something happened in 2020 which made dance productions quite difficult to host. Maybe the kids in the audience don't remember, but we all went through COVID when live gatherings were extremely difficult. And thanks to the incredible work of biologists and biotech researchers, we found a vaccine and we've moved forward, and here we are, gathered together. It seems like it never happened. We've got some masks, very responsible. So just take a minute to feel that gratitude for our health, which is dependent on this cellular world, which the biology community have come together to protect us. So what we're looking at here is something that each cell in our body contains millions, I think, is the correct order of magnitude. I'm not a scientist, but I love this stuff. These are the ribosomes, and the ribosomes make proteins, and we're basically protein robots. So without the ribosome, we couldn't have any of this stuff. They are molecular machines, and they knit. They kind of like my mum, who's in the audience, I think, knits, but instead of knitting delightful stuffies for my kids, they knit the proteins, and they find their way out, and they form everything in the body, pretty much, or a large amount of it. Why is this important? It's because this is how the vaccines for COVID have worked. The RNA vaccines link up with the ribosomes, and they print a special protein, which is part of the COVID virus, which thankfully is enough to train the immune system, but not enough to do us any damage. So these ribosomes team up with biotech, and we have this whole other array of protection, which has enabled us to bring this show back to you. So to celebrate, I've written the dance of the ribosome. So, I mean, it looks quite balletic, doesn't it? You can see the ribosome is attaching there, and there's its weaving. They move. So this is a new piece for tonight, and I'm very excited to have partnered with a dancer, choreographer from Boston Ballet. His name's Michael Stromail. He'll be coming on stage. And our solo instrument for the piece is Philippe Steudlin on soprano sax. Let's welcome Philippe. So, fresh out of COVID, here we go with Dance of the Ribosome. All together, 1, 2, 3. Thank you so much. So we'll be completing the ribosomes journey in future iterations of Cellular Dance, because it's really very complicated how it all works. And I'm not a scientist, but I just love learning these things, and then makes me want to make some music. And well, that's why we're here. So let's see then. We are now heading into the world of developmental biology, and the biology that creates our bodies and our minds, and how the cells emerge with their emergent phenomena to give rise to this complex world we live in. Now, it's a complex world to navigate, but biologists manage this every day somehow. And we're thrilled to have our guest speaker for the evening, who's going to take us on a journey through the development of the inner world of the cell, the cells themselves, and then how the cells together moving in concert, which is our central metaphor. So let's give a big welcome to our guest speaker, Dr. Alexey Varaksa, professor of UMass Boston.
Today, I would like to take you on a short journey to gastrulation. And I want to start by asking you a question. So here we can see 3 fertilized eggs. And can you guess which organism will result after the development of these eggs? This is a rhetorical question, because without additional information, it would be difficult, I think, for you to determine that the top one will give rise to a human, the middle one will become a sea urchin, and the bottom one will become a mouse. And so my lab is interested in this fascinating question: how one cell, through many cell divisions and the process of organism development, gives rise to these beautiful and occasionally intelligent systems. So development is a long process, and there are a lot of steps that have to happen, and these steps are carried out by cells. And so cells multiply. We start from one cell and become billions, if not trillions of cells. Cells become different from one another, and they interact. After a lot of them come together, they have to agree to build organs and tissues. And importantly, they also move to new locations in the organism. In my lab, we use Drosophila melanogaster, the common fruit fly, as a favorite organism to study these processes. By the way, Drosophila starts development from a fertilized egg that you see on the left. Inside this eggshell is one cell that, after all of these processes, becomes an adult fly. And this is a fairly sophisticated process. We study all of these events, but today we will focus on cellular movement. And not just cellular movement, but the process of gastrulation, which is a very important developmental event. This is when an embryo, which at that stage is just one layer of cells, some of these cells move inside and make new tissues inside this developing embryo. This process is so important that a prominent developmental biologist, Louis Wolpert, once noted, "It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." And it will become hopefully apparent why this is the case. So on our journey, we'll move in 3 steps. We will look inside the cell, what molecules are out there that help it move. We will see how cells generate these forces, and then we will see how cells move together to carry out this process. So here, gastrulation involves the movement of multiple cells, but it all starts from the movement of a single cell. So here, you can see one amoeba-like cell that is moving around. And of course, most of us in the audience have played with cats and dogs. Scientists are privileged. Young people in the audience, if you become a cell biologist, you get to play with one cell. So here, this cone-like shape is a pipette with a source of an attracting chemical for this cell. So the cell senses that and moves towards it. The question is, how does it do that? And I need David's help to move the slide forward. Oh, okay, it's happening, I think. Okay, that's good. Technical difficulty. Let me try. Okay, there we go. So, in order to understand how this happens, we have to look inside the cell. The cell is full of different organelles, and the ribosome that David introduced so nicely, and the dancer and the musician, is just one of the many organelles in cells. And some of them are more important for movement than others. Look at this beautiful picture. By the way, look at the credit. David, when visiting the lab, took this on my lab's microscope some time ago. I am not kidding. Yes. See? Look. We have to sell these. Anyway, so inside the cell, you can see structures labeled in color. You can see rope-like structures. These are the cytoskeletal elements. These will be the main players in explaining cellular movement tonight. They mediate the mechanical functions. In red, we see mitochondria, which are the energy source, and nucleus is there to tell everybody what to do. And so these filaments really represent one of the best ways to move things around in the cell. The filaments are comprised of smaller individual protein subunits that can be brought together to build these long, elongated structures. This cytoskeleton is very dynamic. The cells continuously build and destroy these elements. And so you can imagine that if you build a lot of it at one end of the cell, that will push that end of the cell forward. So that's one way to do it. Another very important way is through molecular motors. These are specialized proteins that can walk along these filaments while holding something. And so they can carry various organelles around, and they can slide these filaments next to one another. So they can mediate this pulling motion. All right. And both of these require energy input. This is where mitochondria come in handy. Now, but this is not enough. If the cell just moves things inside, it will just tumble around. What the cell also has to do, it has to attach to the outside. It has to touch the substrate. And so there are other proteins in the cells, some of them are called integrins, that link the cytoskeletal filaments inside to the extracellular proteins available outside of the cell. So we have a complete picture now for a cell in motion. So these are cytoskeletal elements, help it move, the cell is attaching, there is energy source, ready to go. All right, how does the cell generate mechanical force then? We have to look more closely at the kinds of filaments that are there. One of them is called actin. And in addition to forming these long structures, it can form more complex branching structures that look like trees. And in fact, they're concentrated at one side of the cell that generates the movement, and it is called the leading edge. In fact, you can observe these structures. You can observe these structures in an electron microscope, as you can see on the photograph on the left. And on the right, we have a cartoon of what's actually going on. These trees of actin are built at the front, at the leading edge of the cell, and behind they're continuously destroyed. So if you label these structures, you can see a wave of leading edge polymerization of these filaments and then destruction at the back, and you can see these beautiful images of cells generating these leading edges all around. This is great, but this is only one side of the cell moving. What happens if you want to contract the whole cell? Now we have to invoke a second type of filament in the cell called myosin. Myosin is a motor that can attach to actin, and literally it holds on to actin and generates this movement. It can crawl along this actin filament. And if you bind two actin molecules or more together, if they crawl in opposite directions, they can literally generate this sliding motion. This is exactly what happens in the muscle tissue. You can see in the muscle, there are multiple of these myosins oriented in two different ways. They're attached to actins, and it's like, if you're standing like this, I'm pulling the two cables together. This is how the muscle force is generated. It is strong because it takes millions, and sometimes in some people, billions of these molecules to move our muscles together. But what's exciting, at the microscopic scale, a very similar mechanism works in individual single cells. So this is a group of Drosophila cells where myosin is labeled in green. And these cells are dividing. And you can see at the place where the cell is about to divide, you can see a big concentration of myosin. Actin is also there. You can trust me with that. And that helps the cell to pinch into two during cell division. So actin and myosin are really important for that. Now, we're ready to see how the cells move in concert, if I'm allowed this pun today. So, gastrulation. Again, during the development of the egg, there is a stage which you see in the middle, where the embryo is essentially a single layer of cells. And at some point, one side of this embryo gets inside, and some of these cells migrate. And this generates a more complex structure called the gastrula. This is what gastrulation is about. Now, this is a very important step because these three layers, ectoderm, endoderm, and mesoderm, will give rise to all of the cells and tissues in our bodies. So the ectoderm will give rise to skin and nerves, endoderm will generate gut and liver cells, and mesoderm will generate muscle and blood. So it's a very important process. Now, a lot of what we know about gastrulation has come from research in Drosophila embryos. This is a Drosophila embryo undergoing gastrulation. And look, in the fly embryo, this happens on the ventral abdominal side, like about halfway through. Some of the cells decide to move in and form new layers inside the embryo. And so look at the cells located at the bottom. Their outside, or so-called apical side, is facing outward. And what happens is that side has to become smaller and smaller. It contracts. And if that happens along this whole range of cells, that's how these cells move inside. And if you ask me which proteins are involved, you may have guessed correctly, it's actin and myosin. In these cells, if you think of this one cell located at the bottom on the left as a column, there is a concentration of myosin in the middle. And there are radial filaments of actin going from the middle to the periphery of this cell. And at some point, myosin gets activated and it starts pulling from all directions. And so the bottom of the cell contracts. If this happens over a whole group of cells, you get this concerted motion. The cells are moving inside. On the right, there is a cross-section of the Drosophila embryo, and you can see at the bottom, the cells are undergoing this process of apical constriction and are moving together inside during the process of gastrulation. All right, it seems that we know pretty much most things, but there's one more thing that I wanted to tell you about. And the point here is that gastrulation needs to start, and then the cells need to stop properly. And of course, everything during development is regulated, so there is an appropriate signal that comes in the form of this protein called FOG that appears at the right time, in the right place in the embryo. It binds to another protein called MIST, poetically. And that generates a cascade of events that eventually activates myosin in these cells, and only these cells. And that, in turn, mediates the apical constriction. That's how you get that region to go inside. At some point, after this process is complete, there is a molecule that is in flies. It is called KIRTS. which acts as a plug and stops the process. And gastrulation is complete, everybody is happy, the cells can go and do their job building tissues. In my lab, we have investigated this process in some detail, and we published a paper. If you want to know the gory details, you can look up this paper and read about it. But I wanted to show you a pretty exciting snippet from that paper. So we asked what will happen in the embryo if you remove this molecule Kurtz that normally stops the process of gastrulation. At the top, we can see a normal embryo, and myosin again is labeled in green here. You can see that the cells normally only move, this is repeated 3 times, only concentrate myosin in the middle, and then they move inside, and then the process is done. See what happens in embryos that don't have Kurtz at the bottom. In many cells, myosin tries to contract. This happens without order, and the embryo essentially eventually tears itself apart. So you can see how important it is to properly balance on and off signals during gastrulation. And so this is all great, and don't think that this is only applicable to flies. In this movie, on the left, you can see a process of neural tube closure in the mouse with a very similar process where the cells have to zip up and move inside the embryo a little bit. If everything goes well, we get normal development. But if this process gets broken, certain human diseases are associated with it, such as exencephaly, where the brain becomes still open and not properly closed, or spina bifida. On the other hand, in cancer cells, when they get the ability to metastasize, move to inappropriate locations, there is too much cellular movement. So it is very important to study these processes, and this is what we try to model in flies to contribute to this understanding. So we're at the end of our journey. I want to thank my lab. This is us in our beautiful building, Integrated Sciences Complex at UMass Boston. Come and visit. And I want to acknowledge funding agencies, NSF and NIH, who supported this work. And most of the Kurtz project was carried out by former graduate student Fei Chai, who is now a postdoc. And it was a great collaboration with Steve Rogers' lab at the University of North Carolina. Thank you for your attention.
So we're now going to bring up our dancers. And I mean, I hear you've been working together.
Yes, in a bit. We will show you the gastrulation movement.
We're going to show you. So the dancers visited the lab, and they're going to bring them on now. First, I'm going to introduce Meg Anderson, a dancer choreographer, Miranda Lawson, and Haley Day. And you're going to show us something, right?
Yes.
Yes, okay.
Let me see. I hope that this works. Yeah, push it forward. All right, so the dancers are going to show first what happens in normal cells when everybody is happy during normal gastrulation. See, the cells are moving inside happily with one concerted motion. Yes, just like that. Nice and easy, and the embryo is happy. And then we have to see what happens when Kurtz is mutant. The cells are a little bit confused. They're searching for their space. They're not moving properly inside. They're just messed up. Oh goodness, it's stalled. I think it's just sad. So, thank you, and look for these motions in the upcoming ballet.
Did you see my little ball of cells up there? Did she come up on the screen? Maybe later. So you've seen a little bit of our collaborative process. We love to work so closely with musicians, scientists, and dancers. And we're going to perform this journey for you now, cellular dance. And we start with the inner world of the cell and the organelles. We then move to one cell navigating its viscous environment. And then finally, the cells in concert in the fold of the gastrulation. And choreographed by Meg Anderson. And I'd like to welcome our pianist, our solo pianist for the show, William Hume. Where is he? catastrophe. The mutation has halted the progress of the body, and this is something that happens in our lives, and we have to live with it. But thankfully, we have those in our society who spend their lives looking for treatments and cures for the mutations of our body. To take a break in this moment of cellular catastrophe, I'd like to invite up Dr. Chifu Jeffrey Yang. He is a thoracic surgeon at Harvard General, and he's a professor from Harvard College, and he'll be talking to us about his work on lung cancer. Jeffrey Yang.
Thank you. Good evening. What a tough act to follow. I'm honored to join you in this beautiful night of music, dance, and science. Tonight, as we experience the rhythms of the cellular level brought to life by the incomparable composer David Ibbett, I want to take a moment to speak about a topic that gets at the very heart of how our cells work and how we can intervene when they don't work quite as well. I'm talking about screening for lung cancer, and I appreciate the opportunity to share with you about lung cancer screening in the setting of November being Lung Cancer Awareness Month. As we just witnessed through the artistry of this performance, cells moving, differentiating, and communicating, I'm reminded of the magic of how our bodies are composed of trillions of tiny units working in harmony. But sometimes within that harmony, a cell can veer off course. Add together a mutation, a missed signal, a change in the microenvironment, and that cell may begin to grow abnormally, recruiting their neighbors, forming a mass, and spreading. And it can all start with just a single cell falling out of harmony. Lung cancer is one of those conditions where early silent shifts in our cellular world can lead to large-scale effects. But importantly, it's one where we know how to look for those shifts before they become catastrophic. Here's why early detection through lung cancer screening matters. Lung cancer is the #1 cancer killer in Boston, in Massachusetts, in the United States. More women die of lung cancer than breast cancer. In fact, more women die of lung cancer than breast cancer, ovarian cancer, and cervical cancer combined. By the time lung cancer causes symptoms, which can be cough or shortness of breath, it is often spread throughout the body and can be harder to treat. The good news is that a simple, painless, quick and easy low-dose CT scan can detect small nodules in the lung before symptoms appear, when we have the greatest chance to intervene and save lives. This scan takes pictures of the lung, takes less than 2 minutes, and you don't even have to take off your shoes to get it. As a thoracic surgeon, I see every day how profoundly early detection changes the trajectory of lung cancer. When we find a tumor through screening, I often have the chance to remove it with minimally invasive techniques, where you only have a couple small incisions, fast recovery, and most importantly, get a real chance of a cure. Screening opens the door for us to operate when the cancer is still small, and treat it at its earliest stage. Over 90% of patients go on to lead long lives. This low-dose CT scan is safe and it's fully covered by health insurance and recommended by all major medical associations. And yet, 80% of people who qualify are not currently getting screened. Guidelines recommend screening for people at higher risk, for example, people who are currently smoking or have smoked in the past. But we're also studying screening for people who have never smoked. And a Mass General Hospital study led by Dr. Allison Chang will launch the Legacy Trial, which is a screening study for people with a family history of lung cancer. At Mass General, I'm also working with a team on the INSPIRE study, which is a screening study of Black men and women who are at high risk. And there's the firefighter study led by Dr. Leisha Sequist and Erica Warner. Importantly, screening is only useful if we can act on it. Most people in Massachusetts have never heard of screening through low-dose CT scan. November is Lung Cancer Awareness Month, and most people don't know that either. So this is where we need your help, to help us raise awareness and help save lives. Tonight, as we are inspired by the beauty of this music and dance bringing to life the cellular world, I invite each of you to think about how you can be part of this program for early detection. If you or someone you know might qualify for lung cancer screening, Talk to your healthcare provider and encourage your loved ones to do the same. Help us push for greater access to lung cancer screening and help us reduce the stigma around lung cancer. Anyone with lungs can get lung cancer, including people who have never smoked. And help us spread the word that lung cancer screening is real, it works, and is a story of hope. Our team at the American Lung Cancer Screening Initiative, here with us today, we have Kiana, Zach, Jana, Deepti, Brian, and Alex. We're gonna have a table here at the reception area, and we can give you more information or even help you get your screening scheduled. Please come by and say hello, and we'd love to meet you. Our cells are resilient. Our bodies are remarkable. When we give them the support they need with the right screening and the appropriate follow-up, we can intercept disease, restore harmony, and let life's dance continue uninterrupted. Thank you for joining us tonight in celebrating science, music, and art, and for helping transform the silent dance of detection into a choreography of hope. Thank you.
Let's have a huge round of applause for Will Hume, our pianist. Give it up for Will. Let's have the ensemble. Haley Day. Miranda Lawson. Meg Anderson, dancer and choreographer. Have we got Michael? Is he still here? Michael, give it up for him. Philippe Stodlin, our sax player. Come on stage. Jeffrey Young, come join us. And last but not least, Alexei Baraksa. Come on stage, come on stage. Michael, you're here. Here we go. We've been Cellular Dance. And we're ready, 1, 2, 3.